Hello, everybody. My name is Matthew Charles. I'm from the Department of Life Sciences at Imperial College London, and I'm going to be talking today about a project we've had running in my group. We've been really working towards this problem of trying to prioritize the targets of covalent small molecules using an approach that we've called CRISPR-based oligo recombination. And so, for starters, the parasites that we work on and as a model that we're interested in are these apicomplexan parasites. These are all within this apicomplexan phyla and include the Plasmodium cryptosporidium toxoplasma. They cause about 500,000 deaths worldwide per year. And what's exciting about toxo is you're talking about, in terms of seropositivity, around a third of the world's population is or has been infected. So we're looking at about 2.5 billion people. And for me, this is particularly interesting because we don't really know what it's doing to us. For toxo, there's no current decent human vaccines, and drugs in particular available for toxo are limited by toxicity, safety, and low efficacy. And so really we're thinking about these sulfur and pyrimethamine drugs. So yeah, there are other options rather than using these traditional drugs. And so one option that's becoming more popular in the clinic is these covalent small molecule inhibitors. And really, I'd say they have been resurgent, but they've been resurgent probably since the early 2000s. And you can kind of see in terms of publications, they're getting increasing recognition in terms of their success in the clinic more than anything else. And so particularly we're thinking about these oncogenic kinase inhibitors, so afatinib and ibrutinib. And what this has really highlighted to me is that potentially these are a really untapped resource for antimicrobials. If we're thinking about novel and the need for novel antimicrobials, perhaps these covalent small molecules could be a new way to get at this. So how do you start off really? So let's say you want to identify a new covalent small molecule inhibitor. You can obviously screen panels, but the other option is to actually identify sites that you could potentially latch onto with these covalent small molecules. So one way to do this is through reactivity profiling. What the method is you take a broadly reactive chemical probe, you put it onto a biological sample or onto cells, and this will bind to specific chemically reactive amino acid side chains displayed on the surfaces of your protein targets. And what's neat about this is you're using your native protein samples, and so you know these proteins are intact and they're displaying these chemically reactive amino acids handles. You can then use quantitative mass spectrometry approaches and use this as a way to actually discover chemically reactive amino acids. And so what's nice now and really accelerated over the past few years is the availability of different reactive probes for tapping onto different reactive amino acids. And so we have your serine-based probes, cysteine. We can now also latch onto lysines, these oxyziridenes that came out of Dean Toast's lab, and Chris Chang allow you to latch onto methionines. And so these are really powerful tools for actually profiling chemically reactive amino acids across the surfaces of proteins. And so we use this as a starting point to identify chemically reactive cysteines in toxoplasma. And so we use this workhorse-based approach. Well, it's a workhorse in terms of most labs worldwide now in chemical biology, this isotopic tandem orthogonal proteolysis activity-based protein profiling. And so the way this method works is we take extracellular parasites. We can treat these extracellular parasites, initially making a lysate, and then treat this lysate with either a low concentration or a high concentration of probe. And really this is the key point of this assay. Here you're going to have a subsaturating concentration of probe. You're going to preferentially label more reactive sites. Saturating concentration, you will in principle label all sites. You then click on, thanks to a bioorthogonal handle, isotopically or isobarically distinguishable tags, which then allow you to combine these samples for quantitative mass spectrometry and use peptide ratios from the MSMS to actually quantify specific reactivity of different sites. So part of the reason for doing this is that the initial work with human cells and other mammalian cell lines has shown that these hyperreactive cysteines actually are a good predictor of cysteine functionality. And we thought, okay, so we can identify reactive cysteines that have functionality. That's a really good place to then start to try and develop novel small molecule covalent inhibitors. So we identified in toxo about 130 hyperreactive sites. It's represented about 1.2% of the total parasite proteome. You see a distribution of reactivity as one would expect, and a lot of this is actually simply related to specific sensitivity of a given chemically reactive amino acid to the particular electrophile of the probe itself. Looking in a little bit more detail at this hyperreactive data set, we see kind of proteins you might expect to see. So this thyroidoxin-related protein, this ATRAX1, and then proteins we might not expect to see. So the pseudokinase ROP8. So the pseudokinase has a cysteine that sits, a really reactive cysteine that sits on the substrate recognition site. And so then you end up with these lists, right? And so we can do a bit more bioinformatics. We can see that using a phenotype score that was generated from a genome-wide CRISPR screen from Sebastian Laurito's lab, we can see that typically your distribution of cysteines on genes with essential functions, you get about a 30% spread, very similar to kind of all genes in the genome. Interestingly, we see an actual enrichment. So if you have a chemically reactive amino acid, or chemically reactive cysteine in this case, you are more likely to be essential or associated with a protein that is essential for parasite fitness. Using the GO-term analysis to assess this in a little bit more detail, we saw this pretty interesting enrichment for translation-associated components. This was pretty surprising to us. Translation, particularly ribosomes, can obviously be a bit red herring in terms of mass spectrometry, simply due to their abundance. And so we've spent a bit of time looking at this in more detail, and we're pretty confident that this is not actually related to simple abundance, but is actually a genuine result. So the conservation of these guys is spread, as again, you might anticipate. If we look at the conservation across a range of different parasites, including related Cryptosporidium and Plasmodium, but also members of the Candidoplastidae, so the Trypanosoma, we see that there's a range of conservation. What's pretty cool is you see that some are very kind of highly conserved, but only in parasites, suggesting that there are hyperreactive cysteines that are uniquely conserved on clinically relevant parasites. And so in principle, these could be really excellent sites for covalent targeting. Now, one big issue is with these mass spectrometry data sets, which ones do you go after? Okay, so which ones do you try and target? And really this kind of comes down to then the big problem for me. So you have these chemically reactive proteomic data sets, fantastic unbiased systems level approach to kind of identify these reactive handles. But then which ones do you study? And so you have to cherry pick. You then have to go in, you make a conditional mutant. So you'd want to identify a phenotype associated with knockdown of your protein containing a reactive cysteine or site of interest. And then in the background of that conditional mutant, when you've identified the phenotype, you would then complement back with your different amino acid site mutants to say specifically, does this one particular residue contribute to protein function? And as you can, I hope you can understand from that, it's a non-trivial. It takes a long time, and it's also dependent on your success at cherry picking. So this is extremely biased and puts this huge bottleneck in terms of progressing these sites and prioritizing these sites for downstream target-based screening approaches. So could we come up with a better way to do this? So could we come up with a way to actually phenotypically profile the contribution of individual cysteines or amino acids to protein function in cells, and to prevent this early cherry picking and really enable prioritization? And so the method we came up with, we've called CRISPR-based oligorecombinating. It has some parallels with saturation editing that was developed from Jay Shenja's lab. We've developed this method, and we've then actually used it to profile the 75 individual hyperreactive sites in 60 essential genes. And so this is enable expedient prioritization of these sites for the first time. And I'm going to talk you through that data today. Briefly, the strategy that we came up with eventually was that we would target an individual cysteine with a CRISPR-Cas9 plasmid. So Cas9 would be directed to cut on either side using a dual guide strategy of a given site of interest. These double cuts would then promote homology-directed repair with a range of templates that were provided within the same transfection. Now, when one of these donor repair templates is integrated, it destroys the PAM, the protospace for adjacent motif, and also sometimes the protospace sequence. So it means that a given parasite will only receive one single mutation. We put in a range of mutations. So we put in a synonymous cysteine mutation and a stop codon. We're only working with essential genes, and so this will recapitulate, or we anticipate that this should recapitulate the knockout. And then we put in some traditional conservative mutations, alanine and serine, and then a more radical substitution with the tyrosine, as work from the cancer field has indicated that Often a cysteine to tyrosine mutation is associated with a protein-protein interaction destabilizer mutation, so a PPI destabilizer. We generate this transfection, we sample this as a pre-sample, we then follow this through a period of competitive growth, and then sample this as a post. We can then quantify this by next-gen sequencing, and really it's the power of kind of next-gen sequencing that allows you to quantitatively assess the relative contribution of an individual substitution to protein function and ultimately cell fitness. And we were able to benchmark this against the wild type and our stop codon, which again, we anticipate would be the knockout, to really assess how these influence protein function in cells. There was a lot of things to consider, and it's taken us a while to get to this point. I'm going to talk you through a few of these points today, really particularly the gRNA design and how we went on to vector production and mutational templates. So in terms of designing our guides, we actually wrote an entirely new script. We found that the existing guide RNA design programs are basically, they're gene-centric. You put in your gene sequence, you get an enormous panel of guides, and it's quite challenging to identify at reasonable speed good guides to target a given like residue of interest. And so our method allows you to specify a specific residue of interest, specify a nucleotide window surrounding that residue, and then it'll provide a guide output in kind of increasing distance away from that site to really help you prioritize your guides for optimal homologically directed repair. Because we're looking in a shorter search window, turns out your search times are much faster as well. And we found this a really useful tool, and hopefully you will too. It's available as a standalone resource and is hopefully user-friendly. For the plasmid assembly, we turned to a high-throughput plasmid assembly method because, again, we're looking at multiple sites, and we have to have CRISPR plasmids to target each one of these skin cysteines. We were reliant heavily upon Twist at this point for their synthetic DNA, as the synthetic genes we used would allow us to generate these guide plasmids using a linker-based assembly strategy, which then wouldn't require downstream sequence validation, which again, in terms of colony picking and subsequent sequence validation, turned out to be a huge time and cost saving for us. And I think that's really an issue of kind of scale, and I would highly recommend thinking about these synthetic DNA approaches. We were able to generate all of our plasmids within about a week, actually, once we'd got this strategy set up. And we think we can now generate on the orders of a few hundred plasmids using this basic assembly method from Jeff Baldwin's lab in about a week. So that's really powerful too. The next thing we wanted to assess after really figuring out a way to multiplex plasmid assembly was, all right, for our system of interest, should we be going for single cut or double cut on either side of our residue of interest? Should we be going for a single strand or double strand repair template? We focused on a non-essential protein called SAG1, as it's a relatively well-known marker protein for toxo. And we were able to see that we had significantly improved levels of integration when we used a double-stranded template. Likewise, we actually had improved integration when we used a double cut strategy, which again, I think for us is pretty cool, because the double cut really pushes us towards or biases us towards integration in the absence of a selectable marker, as otherwise these mutations are going in silently. So then for our first demonstration with a gene off our actual reactive cysteine data set, for a positive control, we turn to this ISPH protein. So this isoprenyl biosynthesis protein has a reactive cysteine that sits in the catalytic center, and this protein is essential, and so we anticipated the cysteine should also be essential. We get an integration-specific PCR product, so that was reassuring that we have the method is working. And then we actually quantify this for the next-gen sequencing, with next-gen sequencing for the relative distribution of the mutations. As anticipated, we see really strong correlation that any mutation is basically recapitulates a knockout. This was pretty astonishing for me, as these are biologically independent replicates done different days of the week. And so it seems to be a really robust method as well, which was quite reassuring. For a negative control, we looked at this myosin light chain protein. So this protein has two cysteines that are known to be palmitoylated. And actually, these proteins, these cysteines have recently been shown to not be contributing to protein function, despite the protein itself being essential. And this is indeed what we see fall out from our data set. So we can introduce any of our mutations and we don't actually see any change in terms of the parasite fitness. And I think that's what's really cool, is we actually see this direct correlation of protein sequence to cell fitness. So we then did this for all of our sites and managed to achieve 100% coverage for all of our 74 individual sites. And then we were able to do the next-gen sequencing and the quantification of the relative read outputs for these different sites. And we see a range of distribution and different kind of profiles. And if we just kind of zoom in and think about these as more of those histogram plots, think about them as almost like a biochemical fingerprint of functionality, or at least that's how I tend to think of them. We see that some have clear loss of function, regardless of the mutation. Some seem to be site-specific, or substitution-specific, I should say, indicating that there is specific biochemistry associated with that site that is more functionally disruptive than others. In some instances, we've seen gain of function, which again, I think is interesting and an anticipated benefit of this particular screen. And we see, for the most part, actually, that most sites seem to have no contribution to protein function in cells. And so again, we don't really seem to think there's a specific, at least in toxo, a specific association of functionality with reactivity. As you might anticipate, if we're thinking about these residue switches, substitutions according to like a substitution matrix, so Blossom 62, the more radical the substitution, the more likely you are to be deleterious. And then we took a different view and we're like, okay, so then what's the distribution of protein types within these different groups? And so what we see was pretty interesting. So similar to what we were initially seeing with the chemical proteomics, for these deleterious associated substitutions with the GO term, the gene ontology enrichment analyses, we're actually seeing an enrichment in your translation-associated proteins. And so we looked at this in a little bit more detail, and what we found is that the majority of these sites are actually distributed across the ADS ribosome. A lot of them are conserved with related parasite Plasmodium falciparum, and some of them were actually absent from humans. And so, as I said, they're distributed across the surface of this ADS ribosome, and we see a range of behaviors in our core screen readout. And so that, as I said, some were kind of uniquely sensitive to this tyrosine mutation. We did find a couple that were non-essential. What was particularly cool for us is that this suggested that potentially we had cysteines of functional importance associated with apopcomplexan translation. And so could this be a novel route for developing a covalent inhibitor for this really excellent, or at least protein translation has fantastic track record as a targetable process, right? But covalent inhibition is not available as a modality. So we took advantage of the fact that a lab next door, Jake Baum's lab, has this in vitro translation assay set up for Plasmodium falciparum, and Fabio Fischer, an excellent PhD student in the lab, helped us run this assay. We took the Plasmodium falciparum lysate, IVT lysate, and really assessed the capacity of the lysate to successfully translate protein in the presence or absence of a broadly reactive cysteine covalent cysteine alkylating reagent, so this iodoacetamide. And what was pretty neat is we see that the parasite lysate is uniquely sensitive to inhibition by this very broad, it's very small, super reactive. And so actually this is almost somewhat reassuring. So we see this hyperreactive small molecule is only inhibiting the Plasmodium, the parasite lysate, does not inhibit the human lysate. So again, indicating that potentially cysteine targeted covalent inhibition could be a novel therapeutic modality for these medically burdensome parasites. And so what I want to leave you with today as a bit of a take home is that we think that these chemically reactive amino acids are kind of attractive sites for covalent drug discovery, and this has been known for some time, and that we've actually now advanced this cross-disciplinary platform that allows you to both initially identify, but then critically prioritize these sites according to their contribution to protein function in cells, which we believe really addresses this early bottleneck and reduces and in fact removes the need to cherry pick your targets. So you can actually prioritize according to confirmed contribution to protein function in cells. And as this is simply based really on CRISPR, and a kind of solid next-gen sequencing readout, we believe that this is also system independent if your system is accessible to CRISPR. And so hopefully we'll be able to be applied in a range of different disease contexts. And so I just want to leave you now with the acknowledgements. You can check out this work through our bioRxiv preprint and reach me for any email and conversation about the work we've done. It was mostly the work of this fantastic PhD student in my lab, Henry Benz. I'd like to thank our funders, obviously War Society, BBSRC, and Wellcome Trust, and all of our collaborators scattered across the globe. We've had a wonderful time doing this. Thank you very much for the organizer of the event, and I'm very happy to take any questions.